Hello dear students, welcome again. In this slide you can see it is very beautiful because of the flowers. These flowers are used in various occasions like in the festivals, in the weddings, in the cultural activities, in the religious rituals, and these flowers have become so important to us. They are used in every important part of our life. Even to convey the feelings, to show the emotions, we are using the flowers the to show the love, sadness, grief, or to more, we are using these flowers as the symbols. So flowers are making our life very colorful, and besides this, they are also adding a lot of economy to the country. They are adding a lot of tourism to the country. If you see here, there is a cherry blossom seen in the Japan, which is attracting a lot of tourists all across the globe. And we also told about Nilakurunji, which is a Strobilanthes kuntiana, which is attracting a lot of tourists when it is blooming once in 12 years. So it is adding a lot of aesthetic pleasure to all the people, and it is providing a lot of values like social values in the economy values, and it is providing a lot of employment opportunities also. So flowers have become a lot of it is becoming a very important part of our life in in every manner. But flowers, do they flower only for us because we are celebrating the festivals or any important occasions, or do they have any purpose? The plants are flowering because they are having an important purpose. The purpose of flowering is reproduction. So flowers are reproducing parts. They are the reproductive parts of the plants, and these are carrying the male and female reproductive parts in the flower. So we are going to study about sexual reproduction in flowering plants today, and in this we are going to see about morphology of flower, and we are going to see about how these male and female reproductive gametes are produced from these flowers. And starting with the flower, how it is the structure of flower, we are going to see how it is structured. Here there is a hibiscus flower, and this hibiscus flower you can see there is a stem, and usually this flower is a modified shoot. So the shoot part is getting modified to become the flower, and here it is stem, and this stem is followed by there is a calyx or sepals, and then corolla, that is petal, and there is androecium and then gynoecium. Androecium is male reproductive part and gynoecium is female reproductive part. In the androecium there is stamen, which is carrying the anther and filament, and in the gynoecium there is this pistil, which is having the style, stigma, and then ovary. These are the parts of the pistil. So this hibiscus flower, you can also try out this in your home, that you can separate out these parts of the flower from the flower, and you can also display them like this, and you can also share a picture that you have displayed using these. And you can also write those names, like you can identify the stigma and write the stigma's name, then pistil, ovary, stamen, and other parts of the flower. So this is from the datura. This is datura flower, which we don't usually find here, but here in this there are very clearly distinct parts of the flower we will be seeing in this. This is a calyx, and this is corolla, and inside there are stamens, and then there is a stigma, and this is a style, and this ovary, and this entirely it is called as pistil. And further moving on about it, we are going to see, once we have understood the structure of flower, we are going to see about the pre-fertilization events. So where is this pre-fertilization event starting, and how is it going to start? So let us recall that pre-fertilization event, the event which is occurring before the fertilization, is pre-fertilization event. So under this pre-fertilization event, we have seen about gametogenesis and gamete transfer. So we are going to study about gametogenesis, how the gametogenesis is occurring in the plants in the pre-fertilization event. So first, starting with the flowering, how flowering begins in the plant. So flowering is beginning with the help of hormones. So we have told this, when the plants they are in the vegetative phase, from the vegetative phase to the reproductive phase, in order to switch from vegetative to reproductive phase, they need the chemicals. The chemicals are nothing but the hormones. So the plants are synthesizing the hormones, and they are sending the hormones to the shoot area where the flower is about to begin. Once the hormones are reaching that place, they are about to start the flowering. You can see here the vegetative phase is converted into reproductive phase with the floral induction. Means there is a hormone which is helping these flowers to begin. From the vegetative phase, it becomes into the meristematic in the inflorescence. Inflorescence in the sense they are producing a small bud before the flowering, and then they are finally producing the flower. And these flower, once it is produced, it is producing out male and female reproductive parts. The male reproductive part is androecium and the female reproductive part is gynoecium. And you can see this picture is very clear here. In the androecium there are stamen. Stamens are consisting of the anther and filament. The anther and filament are present in the stamen, and this anther is consisting of small ball-like structure. You can see here, those are pollen grains. They are very tiny, and we are going to study how these pollen grains are generated out of this anther. On the other side, there is gynoecium, which is female reproductive part, and this showing a pistil, and this pistil is consisting of stigma, style, and ovary, and this ovary is inside consisting of several ovules. Each one is one ovule here, and these ovules are producing out the these ovules are producing out the egg, and these pollen grains are producing out the male gamete. So these are carrying the gametes. So how is this gamete is going to form? So we are going to see how this gametogenesis is forming, and before that, what are the structures involved in the gametogenesis? We are going to study about them. So starting with the male reproductive part, that is stamen, and we are going to see the detailed structure of stamen and also further into microsporangium and then pollen grain. For now, we are going to see about stamen. A typical stamen consisting of two parts. We already told that a stamen is consisting of two parts. The upper part is anther, then the long stalk part called as filament, and this filament and anther makes the stamen. And this stamen is usually attached. This is called as proximal end, and this is called as distal end. This is distal end and this is proximal end. This proximal end of the stamen is fixed on the petal or on the thalamus. We know that thalamus is a kind of platform wherein all the flowering parts are coming up. So either it is fixed on the petal, or it is attached to the thalamus. And you can see a typical angiosperm anther is bilobed. This is bilobed. All the angiospermic stamen are bilobed. Bilobed means they are having the two lobes. Here there is one lobe and here there is another lobe. And these two lobes are having a longitudinal groove-like structure. There is a long groove-like structure which is making into two theca. This is one theca and this is one theca. So this stamen is bilobed and they are having two thecas, so they are dithecous. So a typical angiosperm anther is bilobed and each lobe has two theca. Those two theca are nothing but dithecous. So if you have to explain in two terms, it is bilobed and dithecous. And you know what is dithecous? It is having a kind of compartment here, and these two are the two compartments are there in a single lobe of stamen. So it is dithecous. So this dithecous in that bilobed structure will form tetragonal structure. This tetragonal means having the four sides, one side and the another side, and then this here and this here. So they are forming the four sides and they are forming the tetragonal side. The anther is tetragonal in the structure consisting of 4 microsporangia. The structures which are formed, the cells which are present in this dithecous lobe, the dithecous, whatever the cells present in this dithecous, they are modified into the microsporangia, and these microsporangia are going to produce microspores. We are going to study about this later on, but for now we know that these dithecous are carrying the microsporangia. The 4 microsporangia are located at the end of the corners and 2 in each lobe. Microsporangia becomes pollen sacs and are packed with the pollen grains. These microsporangia, which are present here in the thecus, in the lobes, they are becoming pollen sacs, and these pollen sacs are producing out the pollen grains. and these pollen grains are nothing but microspores. In this picture you can see how this is the cross section of the anther, and in this anther you can see there are four sides, and in these sides of in these four sides the pollen grain are going to be produced here in the pollen sacs. These are forming into pollen sacs, and these pollen sacs are producing the pollen grains. So we are going to learn further about microsporangia and further about pollen grain, how pollen grain is produced and the structure of pollen grain in our subsequent class.